Hello again, everyone, and welcome to the sixth session in our T-Cell Talks event. If you're just joining us, I'm Andrew Davies from the events team here at Scientist.com, and I'll be your host for this penultimate session. Joining us are Lynn Santos and Mike Jones, respectively Vice President of Regulatory Affairs and Director of Clinical Services and Alliances at STEMCELL Technologies. Together, Lynn and Mike will address critical regulatory considerations for qualifying raw materials for successful T-cell therapy development. With that, I'm pleased to welcome our first speaker, Lynn Santos. Lynn, feel free to take it away whenever you're ready.
Thanks so much, Andrew. I'm delighted to be here today with my colleague Mike Jones, and we will be talking to you about qualifying ancillary, and they're sometimes called raw materials, and working effectively with your suppliers. So I wanted to just take a moment and do a land acknowledgement. We are very fortunate to be working from the Vancouver area. So STEMCELL acknowledges that our work spans many territories and that our head office is located on the traditional unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. And again, we are very lucky to be working in Vancouver, British Columbia in Canada. So, some learning outcomes, or otherwise known as the agenda for today, we hope that you will walk away with an understanding of the latest developments in the regenerative medicine landscape. You will understand the key considerations for qualifying ancillary or raw materials for use in T cell therapy development. And you will learn how partnering with your supplier can help you avoid common obstacles in your translational journey. So we'll jump in. And again, my name is Lynn Santos, and my pronouns are she and her. And I will start with giving you an overview of the exciting regenerative medicine market. So these slides you may recognize from the Alliance of Regenerative Medicine Sector Snapshot. And they are a wonderful source for current progress, news, investments, etc., in the industry. So I highly advise you to take a look there. But I've taken some of their content to demonstrate how this area of cell and gene therapy is growing. As you can see at the top, there are just over 1,300 ongoing clinical trials around the globe. And you will see the breakdown. There is a predominant number in phase 1 or early phase clinical trials. There is a growing number in phase 2 and definitely a growing number in phase 3 and beyond. So there's still a small amount, like comparatively, and we know that that is a natural reality of the translational journey. And but it is exciting to see that companies are pushing therapies into late phase clinical trials and onto commercialization. Another key area to focus on in this slide is that there is a growing number of allogeneic therapies that are coming through the pipeline. So that is an exciting new trend. Moving to the next slide, you can see that the CAR T therapy leads the way with 49% of the focus on cell types in the industry, and you'll see the varying different other types of cell types that are moving forward. Now they have a focus or an image here on the disease types, and oncology remains the lion's share of activities in this space. So there are other smaller areas of focus, but definitely with the CAR T focus of today, it is good to see that the oncology is taking the lion's share. As I mentioned, there is some exciting progress in this space in the sense of therapies getting across the line and being commercialized. And you will probably have noticed back in December of 2023, there was a huge seminal moment that the very first CRISPR-Cas9 gene therapy was approved by the FDA. And this is a very exciting step forward. We're still at the infancy, I believe, of understanding the potential of gene therapy and cell therapies. We're having a lot of learnings in this space, and but it is really exciting to see something gaining commercialization that is leveraging this cutting edge technology of CRISPR and Cas9. And also from the report, they had some forward-looking statements around 2024, and they had the question, I stole this directly from ARM, is a banner year for cell therapy? And as we have noted from the FDA, they are staffing up their various sections of CBER to focus on the growing number of INDs that are coming through, and other spaces in the globe are also recognizing this growth. And they mentioned that the three trends to watch for is increased effectiveness in treating blood cancer. So as we create learnings from the early phases and early commercialized products, we can bring those learnings forward to, again, increase that effectiveness. Accelerating progress in treating solid tumors, which, again, would be obviously very exciting for the market space. And clinical breakthroughs in treating autoimmune diseases. And I wanted to just clarify that STEMCELL is a reagent provider. So we manufacture and sell ancillary materials, or sometimes called raw materials, that are used in the manufacture of cell and gene therapies. So the cell and gene therapy companies are our customers. So we don't run clinical trials ourselves. We support our customers in running those clinical trials by providing the reagents necessary to manufacture their therapies. And here is a brief snapshot, and it is not an exhaustive list by any measure, but there are several new guidances that are coming out globally to help us understand the compliance and regulatory requirements around cell and gene therapies. Now, I've chosen some of the examples here that are listed here that STEMCELL themselves can derive guidance for the manufacturer of our ancillary materials, or otherwise called reagents, for the use in cell and gene therapies. But these are often targeted to our customers. And as I mentioned earlier, we're really at the infancy of this space. And so the regulators are actually learning with us, and they are coming up with guidances that can help define how to move forward effectively and be successful. The last thing you want is to be submitting an IND or a CTA, a clinical trial application, or an investigative new drug application, and have it shut down at the first step because you didn't understand all of the requirements. So the regulators around the globe are really eager to work with industry to provide clarity there. You will note that some regulatory agencies will give out or send out draft guidances. And that's an opportunity for you or your company or different organizations such as ISCT or ISPE or ARM to gather together and provide guidance back on those specific guidances from the regulatory agency. And that's your opportunity to voice your concerns if things are too stringent or not stringent enough, etc. So definitely take advantage of that. So even with the regulatory landscape as it stands today, the one thing that I can positively assure you of is that there is going to be more changes coming in the days and the years to come. We are going to learn from what we are learning today. There are going to be missteps. There's going to be unfortunate circumstances, as with any novel technologies. And as such, we will learn from them and we will adapt the technologies, and the regulators will adapt the regulations and the guidances. So keep an open mind. Keep an adaptable mindset, because whatever you are doing today, just expect that it will continue to iterate. Now I'm just going to do a couple of slides here to talk about the actual considerations for the qualification of raw and ancillary materials. So I gave you an overview of the progress that is being made in the cell and gene therapy market. And I gave you a glimpse of the regulations that surround that. Again, not an exhaustive glimpse, but just a glimpse. And now I want to just narrow down into the considerations for your reagents that you choose as you develop your therapies. So when you choose a raw material, first of all, number one, it needs to work for you. If it isn't working in your application or meeting the performance requirements that have been developed for your protocol, then that's a problem, even if it is GMP and the highest quality. So that is really, really critical. You have to test it. You need to make sure it works. Then you have to go through a very rigorous process of qualification. And I'll just jump down here to the first bullet here, USP 1043, ancillary materials for cell, gene, and tissue engineered products. This is a wonderful guidance that provides a framework on how to qualify those raw materials. So you will need to gather information to demonstrate the consistency of that product, and the safety profile is going to be very critical. So the regulatory agencies expect you or the end user or the sponsor of the clinical trial to be able to defend the use of whatever cell culture media you chose, whatever activator reagent you chose, whatever cell isolation reagent you chose, whatever cryopreservation media you chose. They are not going to go back to the supplier of those individual reagents to question them on why it is good enough for your use. They will be asking you that question. And you will need to ensure that you can defend that use. So that is speaking to, you need to know that material. Now, that doesn't mean that you need to know the formulation, because us as suppliers need to protect our intellectual property. However, you need to work with us so that you have all the information in hand and in your submission, sometimes as a reference to our master file that has been submitted to the regulators, so that you have confidence that it will be defendable in your application. So do not blindly accept supplier claims. There are no current regulations specific to ancillary and raw materials. So I want that to sink in. So there are no regulations specific to ancillary materials. That means that suppliers such as STEMCELL works with the regulators, with our customers, to define what those compliance requirements are. So any supplier can put GMP on their label, but it doesn't mean necessarily that it is in compliance with the needs that are required. So it is important for you to do your due diligence, okay? And that's why an open conversation and a relationship is required between the end user and the supplier, which my colleague Mike Jones will go into in a little bit. So I have listed here some of the guidances. So again, these are not regulations. Okay, so these are guidances, and they are put in the form of a framework of how to qualify a raw material for the use in a cell and gene therapy manufacturing process. So these are good references for you to start to gather and do your due diligence and gather that documentation so that you can defend the use of that particular reagent. So the qualification considerations, I just wanted to lean in a little bit more on the source. And back in this slide, I talked about the safety profile. That is at the highest level of rigorous review from the regulators. So this is something that you need to definitely pay attention to. And this, when I'm talking about a viral safety profile, you need to understand the traceability of that material. So, for example, if you have a cell culture media, it is manufactured under GMP, so that's a great check. It has a master file, that's great. And it has a claim of perhaps animal origin free. So it's saying that there's no animal components in it. Well, that's great, but that's not quite enough. You need to also go to the next level. So that's the subcomponents of that raw material. So let's say it's a cytokine. So you need to have documented evidence that that cytokine was not manufactured using any animal components. And if it was, which in many cases they do, they need to have documented evidence that they have either had viral clearance processing conducted on them, that they have had adventitious agent testing conducted on them, and in some cases, if it's a bovine material that is used, that bovine material has been sourced from TSC, BSC, minimally risk regions of the world that are namely US, Australia, and New Zealand. So that's a lot to take in that single bullet point. But and if you want to learn more, we definitely have more information on that in a separate webinar on our website. The next that I will cover here is the quality. You need to make sure that every time you receive that reagent, that it performs consistently, that it's safe. So that is critical. The risk, where in the process is it used? Sometimes, for example, a cryopreservation media, that's used in like right before that the material is re-injected into the patient. So there's a heightened scrutiny that is, needs to be considered there compared to, let's say, media that is used to develop a master cell bank, which is really, really upstream in the process. So all of that needs to be built into your risk assessment. And before I pass it on, I just want to mention that I want to emphasize, I should say, that the relationship with the supplier is critical. As I said before, everything is changing. Whatever you know today, from the regulatory perspective or from technology perspective, it is shifting, and it's shifting quickly. And how do you stay abreast of all of that? It is through communication. And so it is critical to have that ongoing conversation with your suppliers so that you can effectively maintain your compliance, so that you can be successful in your translational journey and come up with a win-win solution there. So from there, I will pass it over to my esteemed colleague, Mike Jones.
Thank you very much, Lynn. Hello, everyone. My name is Mike Jones. I'm the Director of Clinical Services and Alliances at STEMCELL. I've been with the company for just over 12 years, and I've had the opportunity to work with many of our cell and gene therapy customers and support the use of our products as ancillary materials in over 80 clinical trials so far. So I'm going to share a bit of a supplier's perspective on some elements that make these relationships effective and successful. And I'm going to start with what might seem like an obvious point, but I think it's an important one nonetheless. So, and that is that relationships between cell therapy developers and raw material suppliers should be built on trust and clear communication. Ideally, you are going to be working with your suppliers for years to come. So it's really important to set a strong foundation for that relationship. There are things you can do to make that easier, like putting confidentiality agreements in place to allow more transparent discussions about sensitive topics. And there are also other things you can do to encourage the right kind of environment. One of those that I'd like to highlight is sharing your patient-focused mission and vision with your critical suppliers. From a supplier's perspective, this gives us a sense of how our reagents or our products have an impact in the real world in the context of your workflow. So it gives a strong sense of purpose. It also reinforces the importance of the supplier's responsibilities. And I can say from my time at STEMCELL, there's very little that's more impactful than when a customer takes the time to share a success story about their patients treated with candidates manufactured using our products with us. It really is very impactful. It's also important to establish a clear mutual understanding of responsibilities. So Lynn mentioned a couple of critical guidance documents, USP 1043 and Pharmacopoeia Europa 5.2.12. Those lay out qualification frameworks for ancillary or raw materials, and they touch on some of the things that developers and suppliers can do as part of that process. This table lists some of those responsibilities. So the developer, for example, is responsible for determining whether the material performs well in the workflow. The supplier is responsible for providing quality and traceability documentation upon request for the material. And the developer uses that information to conduct a risk assessment for this material. Now, I won't go through this full table. I think the most important thing to highlight is that, as Lynn mentioned, it is always the CGT developer and eventually clinical trial sponsor who is accountable and responsible for demonstrating to the regulators that a particular material is safe and effective in the context of their specific workflow. Now, ideally, you will be working with GMP ancillary materials that were designed to be fit for purpose in cell and gene therapy manufacturing. And there are many of these available now, especially for T-cell immunotherapy workflows. However, very few workflows are 100% GMP. So you may find yourself using materials that were not intended for ancillary material use or are not manufactured under GMP. And in those cases, you can engage with your suppliers to see if they're able to bridge any compliance gaps that might be present as part of a custom service. This table shows some examples of controls that are standard for GMP products, like certificates of origin, showing traceability to the secondary raw material level, sterility, mycoplasma, and endotoxin to compendial methods, etc. These are things that are standard for GMP products, but often not for non-GMP products. However, you can engage with your supplier and evaluate whether it's feasible to put some of these controls in place as a custom service, along with things like custom labeling, packaging, or modified formulations. And an example there would be selecting specific raw materials that have a favorable viral safety profile. Now, once you have your materials locked in, it is wise to put agreements in place with critical suppliers to ensure that you have continuity of supply and of compliance. Some examples there would be supply agreements where you agree upon a forecasting and ordering schedule to help assure supply continuity. Change notification agreements are agreements where if a supplier plans to make a change to a raw material that you are using, they have to notify you in advance, and then you can discuss and plan accordingly. And as Lynn mentioned, it is important to audit your critical suppliers and verify the quality claims that they are making. Finally, for critical and strategic suppliers especially, it is important to maintain open channels of communication, and may even be worth having a regular check-in with your supplier. And that can help provide a line of sight on important milestones that are coming up for you. Things like an IND submission, for example. If you're planning to submit an IND in the near future, that's important for your critical suppliers to know so that they can be prepared to provide you with a letter of authorization to reference their master file, if there is one in place, to provide you with other quality and traceability documentation to reference in your CMC section, and so on. From the supplier's perspective, it's a good chance as well to provide you with updates on production for custom raw materials that you might be using or progress on other client-driven initiatives. So I'm going to wrap it up there, and I think we've left a little bit of time for questions.
Okay, fantastic. Thank you both, Lynn and Mike, for the great presentations today. And with that, we can move right into the Q&A, and let's bring you both on camera here. Wonderful. And time is a bit tight, so maybe we can just launch right in. And the first question here is, how much more relaxed is the ancillary material guidance for early phase clinical trials as opposed to commercial product? And is it okay to not have full GMP level rigor for some materials?
Yeah, I can take that one. Thank you. I would say that yes, there is more relaxation around ancillary materials that we have found thus far from the regulators. And that includes perhaps that the process validation of that reagent hasn't been completed, or the method validation may not have been completed. So it's following, if you note, the guidance from the FDA for GMP for phase one. So we're taking that similar approach, and that has been successful thus far. The one area that we have found that there is very little wiggle room on is the viral safety profile. So the regulators are being very sticky about that, which I had highlighted in the presentation. And as such, excuse me, that is something that you will need to work with your supplier to ensure that you have the proper documentation on your certificate of origin to support an acceptable viral safety profile.
Okay, fantastic. And obviously, these are very important questions. If you do have any other questions or want more detail, please follow up with the stem cell team. I'm sure they would be happy to elaborate. Uh, Another question here. In the face of ever changing technological solutions in cell product manufacturing, how do the manufacturers and the regulators meet each other's needs, all the way from application submissions through approvals?
Well, that is a fantastic question, and I'm not sure I have an answer. I'm going to give you my opinion, and that is that they are working very closely together. There's many forums, such as ISPE, ISCT, ARM, as I mentioned in the presentation, Bioforum. All of these organizations are working with the regulators to come up and develop with standards and guidances and requirements for the manufacture of cell and gene therapies, and even in some cases, all the way to ancillary materials that stem cell manufactures. So it is a learning curve from both the market as well as the regulators. So please, I would say that if you are a sponsor of a cell and gene therapy, is to open that communication with your regulator in your global region to ensure that you're staying on track with what the requirements are.
Okay, wonderful. Another question here. In your experience, what is the most common issue that you see that slows a project down?
I can take that one. I think from our perspective, and this is thankfully becoming less common, what we've seen a number of times over the years is for clinical trial applicants to go ahead and submit their IND or CTA without having first gone through that ancillary material qualification process. And in those cases, the regulators will typically come back to the applicant with a number of questions around their material controls that require the sponsor to go back to their suppliers and gather more information, and sometimes even put additional materials controls in place. So that whole process can take a lot of time, and the IND remains on hold all the while. So it's obviously not ideal. But I think over the years, we've seen it becoming more and more standard for sponsors to carry out a robust risk assessment beforehand and make good use of pre-IND meetings with the FDA or equivalent elsewhere, and so on.
Okay, fantastic. We are getting a bit tight for time. Maybe we can squeeze in one more, possibly two. Um, can you give us some examples of challenges that Stem Cell has helped previous sponsors, clients, customers overcome?
Yeah, I think I can take that one too. So yeah, stem cells worked with customers to support the use of our products in nearly 90 clinical trials so far, and most of those have actually been with the use of non-GMP products. So it's most common in those cases for customers to have additional requirements in terms of how the product is tested, sometimes how it's manufactured, and what grade of materials are used. And we've been successful in working with the sponsors to put customized solutions in place to address those gaps. And we actually have a dedicated team at STEMCELL called the Services for Cell Therapy program that's available to work one-on-one with clinical trial sponsors on those types of solutions.
Okay, wonderful. Unfortunately, we are out of time here. Lynn, I know you need to jump across to the panel discussion as well. Thank you very much, Lynn and Mike, for sharing your insights. It's been a pleasure having you both with us.